Thank you very much for the introduction, and thank you all for joining here online today. So I will show you what we have in skin sensitization in terms of replacement of animal testing, what has been validated at the OECD as test guidelines, and then why we now need what we call defined approaches to combine these different methods. I will go into the specific defined approaches being validated at the OECD and the dispute how you validate against which reference data. And I will also show you some recent development, how we now also can subclassify potency. And finally, wrap up with the status where we are in terms of full replacement of animal testing. So in skin sensitization, we have the two questions to solve. We need hazard and we need risk assessment. Working in the cosmetic supply industry, of course, risk assessment is of key importance for us because we can still use skin sensitizers in cosmetic, but we need a quantitative potency data to have a point of departure to then do risk assessment. On the other hand, we also have to register our molecules under REACH, and there we just first need a hazard identification, whether a chemical is an H317, and then we also need now to do subcategorization. So we have to discriminate the 1A, the strong sensitizers, from the other sensitizers. And the OECD guidelines, they are here to guarantee the mutual acceptance of data. And that's why for this task of hazard identification and subcategorization, we really need accepted OECD guidelines. While for risk assessment, we need a transparent and reliable scheme, but it does not necessarily need to go through international standardization. So in this presentation, I will only cover the work towards the OECD guidelines for hazard and subclassification, but we have elsewhere also shown how we apply the same tests in a more granular approach when we want to do risk assessment. So there was a lot of talking about the adverse outcome pathways in recent years, and actually skin sensitization was sort of the poster child for that, with the first OECD accepted adverse outcome pathway, which described skin sensitization starting with the covalent interaction of chemicals with skin proteins, and then a number of cellular events, which finally lead to activation of cell proliferation in the lymph node, and then final the skin sensitization status. And the idea is from this OECD adverse outcome pathway, where you have these key events, it could be enough if we have in vitro assays for some of the key, and namely the rate limiting steps in this adverse outcome pathway, and that could then be sufficient to make a prediction. And so far, there are three so-called key event-based test guidelines. One is for the covalent interaction with cell proteins, and then there is one for the activation of keratinocytes and inflammatory signals in keratinocytes, and then the induction of cytokines and surface markers in dendritic cells. These are the three key events which so far are covered with three OECD test guidelines for C, D, and E. And each of these three test guidelines comprises now 2 or 3 different methods. So there was quite a big rush of validating and developing tests in the field of skin sensitization recently. And in general, the validation of these assays at the OECD was quite straightforward. And the reason for that is actually that all these assays were not standalone assays. I just quickly show you the 3 assays. So the first is the direct peptide reactivity assay, which basically just checks whether a molecule can react with a peptide and thereby leads to the loss of these peptide signals. So we have a peptide and the reduced signal indicating reactivity. So very straightforward HPLC assay. This is not even in vitro assay, it's what is currently called an in chemico assay, because there's no biological system involved. And then we have the assay which we developed in our lab, which is the keratinosense, which is based on an NRF2 reporter assay, because it was found that NRF2 pathway, which is regulated by electrophilic chemicals, is actually the most common cellular pathway upregulated by skin sensitizers. There were quite a number of genomic studies, and in all cases, this was the key pathway which was upregulated by sensitizers, which is not surprising because we know that sensitizers need to react with proteins, and we know from the NRF2 pathway that we have this reaction, this reactive surface of the KAP protein, and reaction of a chemical with the thiol molecules on this protein leads to dissociation of NRF2, activation of the antioxidant response elements. And in the keratinosense assay, this is then coupled to a constitutive promoter and the luciferase gene. And so it's a very simple readout measuring the light in a dose response, usually at 12 concentrations in parallel to a cytotoxicity measurement. And actually, induction, a significant induction of the NRF2 gene in absence of cytotoxicity is what rates a chemical positive in this assay. And the third key event covered in 442E looks at a surrogate of Langerhans cell activation. So here it's not really Langerhans cell, but it's a THP1 cell culture, which is dosed with chemicals, and then the induction of surface expression of some key maturation marker, CD86 and CD54 is evaluated. And this is what's called the HCLAD test, the human cell line activation test, which has been first published already 14 years ago, and which is the first test which made it into 442E. So as I have said, all these three assays and the following assays, they were quite straightforward to validate, because the guideline says you can accept positive results on their own from this test to classify a chemical, but you cannot really use the assays on their own. You need an integrated approach for testing with other information from other key events. So that's basically the limitation stated. So we don't have standalone assays, and we need a combination of data. And there were quite a number of publications how data can be combined. But finally, that we have really mutual acceptance at the OECD level, also this combination should be validated. Only then we will have a global replacement. And so there has been now a project at the OECD for, I think, 3.5 years to make a performance-based guideline which includes agreed defined approaches for hazard identification and optional also subclassification. Now, first of all, we have multiple tests and how to come to a final conclusion. So this needs to be a very transparent, simple, and unambiguous system. And there are two quite simple proposals in that draft guideline now. One is what we call the 2 out of 3 approach, which basically is a simple weight of evidence principle. If 2 of these assays are positive, a chemical is rated positive. And if 2 of these assays are negative, a chemical is rated negative. This just makes a weight of evidence without a very specific mechanistic link between the assays. And similar also, the other, what is called the integrated testing strategy, is also a sort of a weight of evidence. But while in the 2 out of 3 approach, we just take the validated prediction model from the individual assays, the ITS transforms the raw data of the assays into scores and then makes a sum of scores, and the sum of score then defines whether it's a non-sensitizer, a weak or a strong sensitizer. So, but here it's also sort of a just quantitative weight of evidence approach. Now, for validation, while I said for the individual tests, the predictivity was important, but not so important because these were not standalone assays. Now, for the defined approach, the reference data becomes very important because it should be validated performance-based, meaning what is the prediction of the final defined approach. So selection and curation of reference data has become very important, as Nicole has already mentioned in a more general view for all these toxicological validations of alternative methods. And then the question comes, which are our reference data? And here we have different choices in skin sensitization because we have the local lymph node assay, which is the animal test, which has a high appeal because first of all, it's a standardized protocol. Secondly, it's the current OECD guideline. And third, the regulators are very used to the LNA. So if we just replace the LNA with something which is similarly predictive, that's very, very much wanted. But there are also cons to that because the LNA actually is itself already a surrogate, a replacement method, which doesn't measure sensitization, but just measures cell proliferation in the lymph node. And secondly, there is an issue that the OECD test guideline states dose selection criteria which have never undergone validation, which makes the test very sensitive but not very specific, as I will show. And on the other hand, we could directly use human data because here we would directly measure sensitization, and the human data actually tell us what we are really concerned with. But the problem there is that the protocol is not so standardized. There are heterogeneous data. It's not an OECD guideline. And sometimes there's the question whether the maximal test concentration was just too low to conclude on negatives. So what the OECD group decided is to do a very detailed review of both the LNA and the human data. I show you now some results for the 2 out of 3 assay, because that's actually the assay which we have together with BASF, the approach we together with BASF have worked a lot on, and which integrates all the 3 assays. So this simple weight of evidence was first published in 2012 with 50 chemicals. And here I show you how the performance versus human data. Later, together with BASF, we published a larger data set on 100 chemicals with still quite similar balanced predictivity around 90%. The team of Cosmetics Europe, together with NICETM, then published a larger data set where the sensitivity went down to 80% and specificity was a bit lower also. And then this data set, which was in that Kleinstreuer Cosmetics Europe publication, this human data actually come from Basket of 2014. We then compared them against the LNA data review of the OECD, which then gave a bit of better predictivity again. And we did a further analysis where we looked at chemicals which are just in the borderline range, in the statistical borderline range of in vitro data. When we exclude those less certain chemicals, these are 18 chemicals, then the actual balanced accuracy goes up to around 90% again. And the OECD then also reviewed all the human data. And the problem there was that the data the data set became quite small, 65 chemicals and relatively few negatives, because very stringent data curation criteria were applied. But what you see interestingly in the last 2 lines, with this reduced data set, we have a very, very similar predictivity as with the larger and less curated data set. So this has now just been published, this draft guideline, including this predictivity figures. And this just shows you the different evaluations done over time. So versus human data, overall, all these assessments came to quite similar predictivities. And what is important, it's quite a balanced accuracy, which is balanced because sensitivity and specificity overall was always quite similar. Of course, as I said, comparison versus LNA is also very important, especially to regulators when it should now replace the LNA test. So in this first four published evaluations, the sensitivity was always on different data sets very close to 80%, and the balanced accuracy also close to 80%. The predictivity was clearly lower in that specific cosmetics Europe data set and by Kleinstroyer et al. And what was then very important, and in this OECD database versus the LNA, the sensitivity is lower as compared when we look at human data. And this 70-40% was considered not good enough. But again, when we then looked at what are the chemicals which are just at the borderlines of the in vitro methods, we have 34 chemicals which are less certain. And if we only look at the 134 certain chemicals, then we are again at the balanced accuracy above 80%. So the OECD guideline now proposes to only conclude on the clear-cut chemicals, and that more data is needed if we are in that borderline range. What is of course also important, since we have the human and the LNA and the in vitro data, to really look how good is the in vitro, we can also look how good is the LNA, which, as I said, itself is a surrogate and a replacement method. And what is interesting in these same data sets over time, what we see is the LNA is really a sensitive method. It's always between 85 and 98% sensitive, but the longer the data were reviewed from 2012 to 2020, the specificity of the LNA is ever decreasing. And one of the reasons is how the LNA is interpreted in the guideline. And the other reason is that these initial studies on these 50 chemicals were chemicals which were really selected to have congruent LNA and human data, because we wanted a good weight of evidence of the reference data to validate in vitro assays against. Yeah, so this shows you, this last figure shows you how important the reference data are. And I show you one example where we now discuss a lot, where is the blind spot? Is the blind spot in the in vitro method, or is the blind spot maybe in the reference data? And this is an interesting case, because what our colleagues developing the H-clot found very early is that actually the H-clot has a quite limited sensitivity for very lipophilic chemicals, but they only looked at LNA data and found this limited sensitivity. And later it was found also other in vitro tests, they always had a limited sensitivity against the same chemicals, whichever in vitro method was looked at. And then the question was, is it really the in vitro data or is it the LNA? And then we recently looked at what is actually the LNA false discovery rate versus human data. So how many of the LNA positives are false positives, and I had shown you before this low specificity, and actually turned out that the LNA has in the different data sets quite a high false discovery rate at high log P, and it seems that it's rather the animal test than the in vitro tests, which here have a blind spot. But this is now again a discussion, what is finally then written in the guideline on this issue. So with what I showed you before, we can today make a hazard identification. We will see what comes out finally with this guideline. But we also need subclassification, and with the integrated testing strategy, this is directly done with that scoring system. but the sensitivity is not perfect. So we proposed an alternative approach, which is the kinetic DPRA, because what we know is that the molecular initiating event is really this covalent modification of proteins, and it might well be the rate limiting step. And the rate limiting step could conceptually be the key driver for potency. So that's why we developed this assay where we, again, like in the direct peptide reactivity, just look how quickly a peptide is depleted by the test chemical. But here we do it on dose, in a dose response and kinetic over time assessment. And with this, we can actually measure a rate constant, and we look at the maximal rate constant at any time, and then compare this rate constant against the potency in vivo. And we validated this recently in a very extended ring study with 7 laboratories. We had a lot of volunteer laboratories because this potency discrimination seemed to be the last key missing link in this field. And we also tested the whole database of 180 chemicals with this assay, and then did a receiver operator curve analysis to check what would be a threshold. And I may need to say, tell that there's a fixed threshold for the LNA result to discriminate 1A to 1B chemicals. And so we just needed a threshold which best reflects this threshold which is currently used in the LNA. And it's a poor chance in the LNA, it's 2% is the threshold, and here we have a log K max of -2. Of course, this is a completely different unit and something completely different, but by chance it's also the number is also 2 again, and this gives then the best predictivity, so we can detect 84% of the 1A sensitizers, and the specificity is in a similar range on these 180 chemicals. So, and this means that when we now combine this kinetic DPRA with the 2 out of 3, then we are able to make a full replacement based on validated test methods and with validated prediction models. So what is the status? This revised draft test guideline on the defined approaches went into the second commenting round after 2 years of very careful data curation and very intense discussion. This was a very, very intense process. And this one now includes this 2 out of 3 defined approach and the ITS, but it's anticipated that more defined approaches will come in future editions, because now it's the database is there to also validate other approaches. And for this kinetic DPRA, this guideline is also just now in the second commenting round. And next week, the deadline for comments is over. And if everything goes well, these 2 guidelines should be adopted in April 2021. And at least for those chemicals which are in the applicability domain of these tests, a full replacement should then be possible for regulatory testing. So this was a very quick rush through a lot of validations. Thank you.